El instructor sigue sin conectarse a la sesión, ¿verdad?
Sí,
Hello.
hola.
Hola,
Hola. Hola, buenas tardes. Sí, todo bien. ¿Tiene falla tienes poco problema de problema de luz, ¿no? Ahorita todo bien. ¿Puede escuchar?
Sí, todo claro.
Ok, perfecto.
Everything is okay.
Everything is okay. Fine, fine. That's great. That's great. So, again, I would like to ask you guys if you have any doubt from yesterday's session. Any questions regarding the materials or regarding any work or if anybody is interested in also there is one more thing I would like to tell you here that I am organizing one organ on a chip course. And this organ on a chip course is basically, it's like a 5-day course. And in one of the days, we will be dealing with some practical work, minor practical work also, if there is a possibility. So this is a 5-day course which will be organized depending on the number of students. And if you are interested in, then we can get into if there are like more than 2 or 3 students or 5 students who is interested in having this course, so that it's basically what is my work at is we want to have a complete picture of like we want to give an idea, and we want to create um, uh, like some research group in the field of organ on a chip. And if more and more people are interested in, then we can have a proper scientific group. Because see, this particular field needs a lot amount of activities, a lot amount of work. Research activities are required for this particular job. The idea is that we have to formulate. The idea is that we need to make a complete group, because, for example, there is like a breast on a chip, then lung on a chip. There are different kinds of chips which we need to make: intestine on a chip, stomach on a chip, and brain on a chip, heart on a chip. All right. Then blood on a chip. So these are the different kinds of chips which needs to be made. And apart from that, we also need to integrate them, or the integration is also required together, so that once we integrate them, then we can understand how the traveling of the drugs or anything happens, how they are moving from one chip to the next one, to the next one, to the other. And if the chips are moving, I mean to say the drugs are moving, and we identify that, then it is becoming very critical that we understand. So it's not a one man's job. It's like a huge army which is needed for this particular work. So this is the reason I'm also insisting that people, because see, okay, I am doing like to tell you the fact. I'm a researcher, and I will be just doing this kind of work, and uh, like it will take a lot of time, 10 years, 20 years. For me, there is no issue, right? But then you can imagine a patient who is suffering from kidney cancer. He doesn't have that much of time. Triple negative breast cancer, she doesn't have that much of time. They will have it's aggressive cancer, and she needs rapid treatment. In other types of cancers, in prostate cancer, for example, it takes a lot amount of time, 5 years, like a patient can survive 5 years, 10 years. And there are many medicines which have been given to these patients, and the patient can, and doctors also have got time to do some experiments on the patient, right? But then doctors cannot afford to spend time on a patient who is suffering from triple negative breast cancer. So this particular cancer is so aggressive that in within 3 months, the patient dies. So if the patient is dying within 3 months, though, you can imagine how many people might be suffering. At least 5% of Mexican women are suffering from this particular disease, triple negative breast cancer. So you can, out of 15% of the total women are suffering from breast cancer. Out of that, 5% are suffering from this particular triple negative breast cancer. So this is so dangerous disease, and then people all like non-small cell lung cancer, which is also very aggressive. 6 months patient is having time, and then he dies. So we don't have time to wait and sit together and enjoy life. And we it's like that is the urgency. It's not urgency, it's emergency basically. You can imagine there was a patient, like when COVID-19 came. So it was an emergency period, and everybody was sitting at home, and they were just waiting for for a vaccine to come. And then people were working day in, day out just to get this vaccine. And once the vaccine came out, within 3 months or 6 months, people started coming out, started moving out and having their life. So at least in COVID, you had maybe people had what do you call, they could restrict themselves. They used to sit at home because they are well enough to have their food. But this particular disease, cancer, is such a emergent disease wherein people, they don't know and what's going to happen next. And the chances of death are almost like 90%. 10% chances are there for the survival, 90% chances are there that they might die. So that is the situation, and that's the emergency. That's the reason I'm like insisting you guys, who are actually in biomedical engineers, who are biomedical engineers. So if you guys take up the job, take up this particular field, wherein you can integrate organ on a chip with a lab on a chip, have a chip which is integrated with everything, we're having all the systems together, then this will be, I will say that yes, we can have some difference with this particular disease pathogen. Otherwise, this will be on, and people will die, and we will just see and wait and watch kind of situation. So this is the reason I'm instigating you guys, and that is the main motto why I am taking a course in of biosensor, Diplomado de Biosensors, because I think that you are coming from the background of biomedical engineering, so you are the right people to work on this area, rather than anybody else. A nanotechnologist also, if he is a bio nanotechnologist, it's okay that he might be able to work. But biomedical engineers, they are well aware of the physiology, body physiology. You're well aware of the structures, and then you're well aware of how the machinery works, how biosensing system can be designed, and all the connections. Every electrical circuits can be connected to this organ on a chip model. So everything is like well planned for you guys. So I feel that's the main motto that why I'm teaching this course, and I would like to help you to get into this. I will be just you can call me a torch bearer. I can just show you the torch that this is a field, this is the way, but actual work will be done by you guys. So that's something which I want to tell you. And if there is any possibility that you can join this army, it will be great. All right. So next we can start with the. So do you have any questions first? I would like to ask you guys, and then we can move ahead with whatever we are planning to. Please, if you can ask any, if you have anything, any doubt, please ask me. Anybody? Hello. Any questions? Do you have any questions? You guys, can you hear me?
Creo que no hay dudas.
No hay dudas. Okay. Ok, hay algunos otros gentes que tienen dudas, algunos otros estudiantes que tienen alguna duda. Ok, yo tengo pocas dudas. Ok. Uh, I would like to ask you guys that why there is a need for the development of organ on a chip. Can anybody answer this question? Why there is a need to develop organ on a chip models? Hello. I can ask somebody. Maybe Sergio. Sergio, eso es mi pregunta aquí porque nosotros queremos, oh, perdón, tiene chat. Ah, ok, no. So, mi pregunta es que usted conoce qué es de razón para utilizar organ on a chip. Nada más 3 minutos y ya terminamos con este. E iniciamos de otro parte. Segundo tópico. Sí, Sergio. ¿Usted tiene alguna respuesta, por favor? Ok, no es Sergio. Sergio, yes, tell me. I cannot hear you.
To detect, si hay enfermedades, podríamos
Ah,
detectar con un lab, una chip.
ok. No entendí, perdón. Otra vez, ¿puede hablar?
Ah, ahí ¿sí me escucha?
Sí, yo escucho perfectamente, no hay problema.
Early detection of, ah, hay enfermedades.
Ajá, ok. Early
Eh,
detection of enfermedades, pero es lab on a chip. ¿Qué es de razón para organ on a chip? ¿No? Ok, ¿hay algún otro estudiante? Francisco Josué Portos, ¿se puede hablar? ¿Qué es de función de organ on a chip? ¿No? ¿A Isaías? ¿Isaías también? Sí, puede hablar. Uh, Isaías Flores? Ok.
¿Me oyen? ¿Me oyen?
Ajá, ok. Tell me.
Yeah. Sorry, I missed the question. Could you
What is the
please repeat
function
it?
of organ on a chip? Why do we need this? What is the need of organ on a chip? Why there is a need of developing this organ on a chip?
So we could have a better understanding on how cells do their job. We can study in a deepest way. Maybe, for example, what I remember yesterday is that we can have many advantages on diagnosis and how to develop more. For example, if we get a signal that we want to check, and also for cancer cells, because yesterday we mentioned that in biomedicine. Uh, for me, that's the only part that I remember really at this time. Okay.
Mhm. So the thing is, like you could remember that if we are having a drug and they will be passing through different phase clinical trials, like clinical trial 1, 2, 3, 4, and uh, and then what happens is if we have the studies at the end, because in the clinical trial 3 and 4 we will be actually dealing with human samples or human patients, and by the time it reaches to phase 4, clinical trial phase 4, then the company, the industry, the pharmaceutical company has invested almost billions of dollars. And if it has invested so many billions of dollars, then it is quite imperative that the drug must be accepted and cleared. And if it is not cleared, the company loses that much of amount. And that is one of the reasons also that industries are, pharmaceutical companies, most of the time they don't come up with a patented drug. They will simply take already patented drug from other companies, and then they will modify that, and then they will come up with another drug, so that the research thing will be avoided. They don't have to do a lot amount of research. And that's the idea. That is the main motto. Why industries are interested in not getting into all this business of investing too much money and investing ideas, and doing too many work on identifying the drugs like that. Hmm? So that is the main motto. Hmm? So this is something which is very critical, which people are mm, avoiding nowadays. That's the only thing. But why? Most of the time, these sensors are really very critical. I mean to say, these drugs, they are very critical, um, and at the end of the clinical trial 4, most of the time, sorry, in clinical trial 1 and 2, they most of the time they study with animals, not the human beings. So when they reach to the level of animals, sorry, human beings, then at that time most of the allergies or most of the reactions happen, and because of which the FDA will disapprove the drug. So that is a major setback for them. So this is what why we have to like work on those lines. Okay, and now we will be working on studying this genosensors or DNA based sensors. Mhm. Mm. I'm just opening the sections. Just give me a minute. Mm. Okay, now we will start with a new topic today, which is dealing with genosensors. All right, now what are genosensors? Can anybody tell me what are genosensors?
What? Sorry?
What are genosensors? All right, I will share with you. Now let's do a group activity. Okay, let's do a group activity here. How many students are existing here? There are 14 students, right? Including me, there are 15. So there are 14 students. So what we will do is uh, uh, we will make a group of 4 students each. And I'm sending you one article right now. Okay. And I want you guys to read the article for 15 minutes and give me an overview. Like you have to explain me the article. Okay. And I'm just sending you right now that work.
Wanda, we are going to choose our team.
All right. If you no, I will select the team. All right. I'll select the team right now because yeah, I don't want to have any confusions. So I'll just select the team for you guys. And there are 2 articles I will be sending. Okay. Okay. Now this is fine. And I'm just sending you desktop article for group 1 and electrochemical genosensors. Okay. I think yes, this one is good. Oh, I'm just sending. Just give me a minute. I'm just sending it. I'm just taking time to download it. Nothing else. Yeah, this one is okay. Article for group 2. Now I'm attaching files to the computer. So I've sent two files here. And so I'm making, so you can see here, there are 2 files, which is for group 1, article for group 1 and article for group 2. So who will be in group 1? I'm just telling you right now. I'm just calculating how many, 1, 2, 3, 4. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. All right. So group number 1, Aranza, Lisbeth. You can hear, right? You can say at
Yep.
least, you can say something because I should know that whether you have heard or not. That's the only reason. Just want to confirm. Aranza,
Yep, I'm here.
group 1. Carrillo, Guzman, Ricardo, group 1.
Okay.
Citlali, Guadalupe, group 1. Dennis Ramos, group 1. Enrique Enciso, group 1. Francisco Josue Portos, group 1. Isaias, group 1. So 7 people in one group. Okay. And then Jesus Humberto Garcia Ramos, group 1. Jose Raimundo Padilla Hernandez, sorry, group 2, sorry. Jesus Humberto Garcia Ramos is group 2. All right. Jesus Humberto Garcia Ramos is group 2. Jose Raimundo Padilla Hernandez is group 2. Jose Valentin, group 2. Luis Enrique, group 2. Miriam Cepeda, group 2. Then Rodrigo Solis as group 2. Sergio Trejo Villegas, group 2. Anybody else is remaining? Hello, guys.
Creo que ya estamos todos.
Now, okay. Now you guys have to create breakout rooms. So I'm creating a breakout room for you guys.
Excuse me, doctor. Did you send the articles?
Yeah, I have sent the articles here on the chat.
I can see it.
You cannot see?
No.
No.
Oh. Now you can see, maybe. Yeah, you can see now?
Yeah. Thanks.
Yeah. So see, I am creating 2 breakout rooms. Okay. And here the participants can choose their room. So depending on the number, like you are in 1 or you are in 2, select your rooms automatically. All right. And now what you have to do, article for group 1. That particular group 1 needs to discuss, needs to read. Okay, now the timing is 7:46. So by the time you enter into the rooms, it will be 7:50. And till 8, you can discuss about the article. You can talk about the article, or you want to come up with some, like you just read it. I'm giving you 10 minutes time. You read the article, and and then you can create a resume for the article. All right. And now these 7 students, what you have to do is, the 7 students, you have to, each one of them, for example, I'll show you here itself. Share screen. Article for group 2. All right, I'll just show you here. So this is article for group 2. And you can see here. So here there are like circulating biomarkers. So circulating biomarkers, so you can divide amongst you 7 guys, the different kinds of biomarkers. 3.1, electrochemical genosensing of cancer biomarkers. So one student can take up that. One student can explain the introduction. All right. One student can explain the introduction. The second student can talk about circulating biomarkers in general. Then the third student can talk about electrochemical genosensing of circulating biomarkers. All right. Then fourth student can explain this electrochemical genosensing of cancer biomarkers individually. So that will be the job of the fourth student. Then the fifth student can talk about electrochemical genosensing of neurodegenerative diseases biomarkers. That will be the job of fifth student. All right. Sixth student will talk about electrochemical genosensing of viral infection biomarkers. Seventh student will talk about electrochemical genosensing of bacterial infections biomarkers, like that. So this way you divide the chapter, the review article into 7 students, and one of them, if one of them can consider, even a seventh student, like one guy can take 2, and the last 7th student can take general consideration. What are the general considerations here? So just read it and then give an overview of that. So I want this thing to be done with you all. So will that be okay for you? Let's do this exercise. That's what I wanted to explain. Will that be okay for you guys?
Yeah,
Hello?
it's fine.
Hello? So I am creating 2 breakout rooms. Everybody has understood what I'm willing to say?
Yes.
Yep.
Perfect, perfect. I have created 2 breakout rooms. So you can select automatically. You can yourself select the rooms. Let participants choose the room. That is the option which I have selected. Sorry, close all rooms. To the main session in 60 seconds. No, no. How to choose the time limit for the group? Oh God. I have selected, yeah. 30 minutes. Fine. 30 minutes, all right. Okay. You can, I think you can start. There is no problem. You can get inside now.
Profe, una pregunta. ¿Cerró los grupos? Es que me conecté, pero me volví a sacar. O no sé.
Sí, sí, usted puede entrar, ¿no? Directamente. ¿Usted
Ya,
está en qué grupo?
en el 2, pero es que ya no me da la opción de unirme. Es que me uní pero luego me dijo que o sea, me regresó automáticamente. No sé si a los demás le pasó lo
Perdón,
mismo.
otra vez, ¿cómo se llama? ¿Dónde es? ¿Miriam o
Sí,
Miriam? Ah, ok, ok. Miriam, usted está
en el 2.
en dos, pero yo puso en. Ajá. Usted está en dos. ¿Qué pasó, Miriam?
Es que creo que, bueno, más bien, creo que, bueno, no sé si a los demás les pasó también, pero es que ya tampoco me sale la opción.
I have to create, open all rooms.
Y mentiría, profe, nunca he hecho eso.
Ah, join. Is it okay for you guys now?
Yeah, we get the alert.
Now you can join.
I think so.
Just check whether you can join. Yeah. Hola a todos. ¿Todo está bien? ¿No hay problema? Usted leyendo, pero por favor puede discutir también cuando usted quiera, ¿no?
Sí,
Sí,
todo
Rey.
bien.
Sí, ok, perfecto. Gracias. Gracias.
Profe,
So you could get to know everything.
no, bueno, es que yo soy del otro cuarto, bueno, del otro, ajá, del otro cuarto, del otro bloque, solo que, bueno, salí a la sala y no estaba, entonces supuse que estaba aquí adentro. Es que
¿Salió automáticamente?
No, no, o sea, es que, bueno, estábamos discutiendo que pues todavía nos falta leer un poco, pero según yo ya pasaron los 10 minutos, entonces queríamos ver si nos podía dar un poco más de tiempo porque todavía no alcanzamos a discutir. No sé si aquí ya pudieron discutir, pero,
Si usted quiere discutir, hay 5 minutos más y usted puede discutir y después puede presentar en frente de mí.
o sea, con presentación y todo.
Sí, usted si quiere, cada uno estudiante quiere discutir, quiere explicar.
Es que, o sea, apenas estamos terminando y todavía no,
Sí, está bien, 5 minutos más y ya.
pero lamento la pregunta, pero entonces quiere que hagamos una presentación, o sea,
No,
con
no, usted no
PowerPoint.
quiere, usted solo quiere hablar,
Ok,
presentar es
ok.
hablar. Nada más, usted que entendí en esta parte, y solo quiere hablar,
Ok, ok, vale. Sí, entonces
nada
unos,
más. Ujú.
puede ser como hasta las 8:20, más o menos.
Sí, exactamente, es 8:20, perfecto.
Gracias.
Iniciamos, ¿ok? Gracias, ya. Otra vez usted quiere entrar en breakout rooms.
Oigan, no sé si quieran ya empezar como a discutir. Según yo había entendido que teníamos que repartirnos en general como todo el artículo, no sé si estoy mal.
Ah, sí, algo así había mencionado, pero,
Perdón,
ajá.
otra vez, Aranza, no entendí bien, perdón.
I'm just talking with my team.
Ok, ok,
Yeah,
perdón. Una gran
I'm
disculpa, no
sorry.
entendí. Gracias, sí, sí.
Entonces no sé cómo quieran que lo, nos lo repartamos
Um,
para.
Podríamos, escoger un tema quien guste. No sé, si ya hayan pasado en uno. A mí me gustaría nada más como que hablar de la parte de los genosensors en VIH. Ya ven que tenemos ahorita los dos de hepatitis, el de VIH. Si pudiera yo hablar también un poquito de eso, está bien, es un tema que está chido.
Pues igual podríamos como hablar cada quien de uno, ¿no? Porque vienen varios. Viene
Sí.
de hepatitis, VIH, coronavirus, adenovirus y no sé qué. Como que vienen bastantes. Como es como un artículo, como de varios artículos resumidos, ¿no? De cómo se usaron.
Ujú.
De su aplicación del genosensor de genosensores en infecciones y virus. Bueno, en virus, enfermedades virales.
Me parece bien, si quieres yo agarro el de hepatitis.
B o C.
O
Yo
las
quiero
dos.
influenza.
Entonces agarro la hepatitis B, la primera.
Yo
Yo
el,
creo coronavirus. Perdón.
yo el de adenovirus.
Y damos como
El
un... Ay, perdón.
sí, fue el de inmunodeficiencia, ¿no? El HIV.
Si quieres hablar también de ese, no lo dividimos sin problemas, como tú quieras.
También falta creo que una
Ah,
hepatitis, ¿no?
sí, es que como ya no escuché bien cuáles faltaban, más bien.
Mm, sí, creo que falta ese de
Mm,
VIH y uno de hepatitis. Y creo que eran dos, ¿no?
sí, es la B y la C.
Escuché que alguien
Ah,
por ahí
pues
dijo que si seguía viva la C,
es esa.
que, justo ya te la cortos.
Sí, creo que Denise escogió la C. Ah,
Si quieres nos reportemos ese de los del de VIH. ¿Qué te parece, Enrique?
Ah, ujú.
Y
Excelente.
no, también está la primera parte de qué son estos y
Ah,
el
pues sí.
multitarget al final de los casos también por si quieren agarrar esos. Y según yo soy la hepatitis B, ¿no?
Sí, sí,
ah,
sí, sí.
ok, ok. Igual no sé si las partes de, por ejemplo, lo que viene así como cuatro o cinco, dividido como en secciones, esas también, como comentar algo un poquito, no sé. O sea, la introducción sería como decir en qué se basa el artículo.
Sí, sí, sí.
No sé si alguien quiera como, igual y yo podría explicar en qué se basan los genosensores, porque ya viene la parte 2 de genosensores y más o menos dice qué es y como las etapas. Podría hacer un poquito, un resumen de eso y hago el coronavirus. No sé si alguien quiera decirle un poquito de introducción.
¿Te rifas,
Un
Enrique,
poquito
o qué?
Ah, sí, entonces hago la parte del multitarget.
Ah,
Súper.
ok,
¿O
va.
sí? Ok.
¿Quién iba a hacer introducción?
Sí, algo así como lo que acabo de decir es un artículo que habla de varios. Bienvenidos. Algo así.
¿Ese lo vas a hacer tú?
Pero podría. Entonces,
Y que alguien diga la
no
conclusión.
nos falta nada,
Porque
¿verdad?
ya,
No nos falta
de
nada, ¿verdad?
los, de los de eso de VIH y así, no. Pero sería nada más
Excelente.
quién puede como introducir así como de, pues el artículo habla de esto y las enfermedades virales y ya. Y
Ah,
alguien
sin
la conclusión.
problemas. Sin problemas. Sí, sí, sí.
Va, entonces ahí la introducción. Y alguien que pueda decir la conclusión, no sé, alguien que quiera. Las seis.
No se peleen, calma.
Lo de la conclusión.
Sí.
Ah, sí, pues igual como el punto 4 no es mucho, pues puedo, ajá, ver lo de la conclusión.
Sounds great. Ok, va.
Ujú.
Totalmente tuya, no pasa nada. Sí, igual cada una de las partes y ya, a ver cómo nos va. Sí, no, no nos complicamos la cabeza. Sí.
I think we can start now. Close all rooms.
Recording in progress.
So guys, now group 1 can explain me the article. Yes, group 1.
Okay. So please tell me when you can see my screen.
Yeah, you can start.
Thank you. All right, so we are the group 3, and the paper that it's for us, it's genosensors as an alternative diagnostic sensing approaches for specific detection of virus species. This is a paper which contains multiple experiments and testing on genosensors for detection of multiple human viruses, which affects most commonly the human being. So these are the guys who made this possible. And to understand a little bit of our paper, let's start with a little abstract here. As I told you, this is a really good important topic because of the millions of deaths that are annually reported. So here is an explanation that the main virus that it has triggered this paper, it's the COVID-19 virus. When they continued starting these studies, they mentioned that biosensors are really useful for detecting viruses. We have already studied many ways on how to detect these kind of things, and there are for many other examples biosensors which are introduced in here and multiple techniques as well. For example, it's good to know that these genosensors are only good for improving the sensitivity and the specificity. So this is why electrochemical, optical, and SPR genosensors have been applied on these viruses. So we are going to start on which viruses we are discussing here. And first of all, let's move on to the first one. And it says, where is it? Where is it? Well, genosensors. You have already maybe studied this on your own paper. So I will skip this because genosensors, it's based on that detection of DNA, RNA, and many multiple ways are defined to get this goal. So for example, this one here, it's an investigation. As I just mentioned, me and my team has gone into this complete paper, and many viruses have been studied. So first begin, it's hepatitis V virus.
About the hepatitis B, we have the review of 3 different genosensors. The first is electrochemical, and we use genosensors because we need more specificity, selectivity, and rapid detection. So with the electrochemical, we have that, and it's based on thin doped of And I don't know how to say this. En WO3 y N2 o 3 and an alright with the tension limit of 1 FM. El psychoelectrical use crystal quartz and microcantilever biosensor, and it's usual in real time to has a really fast response. And the last is the SPR that is level 3 and has also a really high sensibility and is based on a loop medieval isothermal application and has a limit of 4.494 and for 10 into the less 18 potencia, and the response is just 17 minutes. So that's the different options that we have on Genosensor to test hepatitis B.
Super, thank you, thank you. So let's move on with hepatitis C virus. Great, great, thank you. And let's continue with this human immunodeficiency virus. VIH, HIV. So this virus, it's well known to cause the syndrome named AIDS or SIDA for us in Mexico. So this first part talk about many statistics on which type of HIV viruses we got. We got the first one and the second one. They have their same origins in animals. The first one in chimpanzees and the other one is on monkeys. So mainly on the world, it's most spread the first one. So most of the genosensors that are discussed in here are related with the first one. So yeah, this is good to point out that the first alternative to treat this first disease is antiretroviral therapy, ART, and unfortunately for this therapy, most of the humans have no access. Around 46% of the people which has already HIV doesn't have um, an opportunity to get this therapy. So that it's the desire to develop more genosensors for this, okay? The most used techniques that we know for HIV, it's immunosensors, which based on immunotesting. And the other ones that we are, that it's part of this topic, genosensors. Here there are four techniques which are explained. The first one is the electrochemical one. Okay, this technology also use maps. Okay, to monitor the HIV genome sequences. Okay, so this is what we are going to detect. Okay, for example, here in this part, it's the same thing, right? It's just mentioned for HIV, not for HIV-2. Okay, so the first one, the electrochemical one, it's an probe which has absorbed the graphene nathium modified glassy surface, okay, from carbon electrode through some type of bonding, okay, PP bonding. This it's a good one for detection as the single-strand DNA probe with the hybridization. It forms a double-strand DNA, which can be detected on the electrochemical sensor. For this, we got limit detections and a linear range, which are really low. -10, this is picomolar, so it's really good to have this type of detections. Again, this is another one electrochemical sensor, which is a little bit more complex, but it's the same, right? We got an electrode which wants to detect this single-strand DNA. And for that, this whole complex is used and it upgrades the sensitivity and the selectivity, okay? There is another one, which is like a piezoelectric biosensor. It's based on a potential microcantilever genosensor, okay? When the genosensor it's detecting the IHV relies on the layer, and then this layer it's bonding, okay? This bonding, it's the signal that we are going to detect and to identify if we have the DNA that we want, sorry, okay? This one was the most interesting one. And last but not least, there is another one that needs the SPR, sorry, surface plasmon recognition. I don't remember correctly. But yes, this was used as the same. They had immobilization on a gold sensing chip. And then this was used to detect how the sensor was how was working technically, okay? So Yeah, this is all for HIV. Let's move on to human papilloma virus. All right, maybe I guess we can move on to the next one. So, influenza viruses is the next one.
Okay, in the next part, the article tells us about influenza. They told us that in recent years, a significant advance had been achieved for the detection of influenza virus, and like virus isolation via cell culturing, shell via culturing, serological assay, and polymerase chain reaction. But it has its limitations, like relatively long detection time, expensive instruments, sample handling, post PCR analysis, and requirement of well trained technicians. And the article told us that in recent years, there had been advances in biosensors for virus detection, and they have led to the development of simple, rapid, and sensitive biosensors for the detection of influenza viruses. For example, we have the label-free detection method for the type A influenza virus by using carbon multi-wall nanotubes, and the result showed that this biosensor can reduce the response time required for influenza virus type I to 4 minutes. We also have an electrochemical biosensor by using DNA. A tetrahedral nanostructural proof was immobilized onto the gold surface and was developed to detect the hemagglutinin gene of influenza. And we also have an electrochemical biosensor by using... no, creo que son todos. Ya.
Super. Okay. Okay, let's continue with coronaviruses.
Yeah, I'm going to summarize the application of sensors for the detection of coronavirus. First, sensors are widely used for virus detection, particularly the SARS-CoV-2 virus responsible for COVID-19. Various approaches have been developed to enhance sensitivity and speed of detection. Among these, sensors combining plasmodic photonormal effect (PPT) with LSRP, yeah, detection transduction, stand out, enable highly sensitive and reliable detection. And additionally, efficient detection has been achieved by combining a lateral flow-based sensor like LFB with multiplex reverse transcription loop-mediated isothermal amplification, and with a detection limit of 2 copies of the target peer reaction. Also, a colorimetric biosensor based on gold nanoparticles has also been developed to identify the SARS-CoV-2 and a nucleocapsid protein gene from RNA samples with a visual naked eye response. And also these advances in virus detection, particularly this coronavirus, are crucial. Well, they mentioned that are crucial for organizing surveys and control of the pandemic in well at that moment in the past.
Super, super, super. So let's go to multi-target detection.
Mhm. In this part of the document, the article focused on the simultaneous detection of multiple viruses, highlighting the importance, the growing and the interest in the development of the biosensors capable of diagnosing different viruses from clinical samples at the same time. Because this strategy provides multiplex virus genome detection with the lowest price and time wasting, which is an important health issue for all the nations. Um, the it's mentioned three works, for three examples, like one SPR genosensor with a gene chip technology for the simultaneous detection of the genome of nine respiratory viruses, including influenza A and B, H1N1, PIB, RSV, adenovirus, and coronavirus. In less than 3 minutes. The sensor demonstrates high sensitivity, specificity, and reproducibility. Another example is a multifunctional printed molecular sensor for the simultaneous detection of hepatitis A and B. And this is this biosensor had a difficult because the structures of the viruses are uh, similar in the external structure. And the biosensor prove specificity. And the last example that mentioned, it's an electrochemical genosensor to monitoring three common HPV types. And the genosensor show efficient detection with the specific detection limits from each HPV type and minimal cross-reactivity on non-specific binding between target viruses and probes. The point 5 only mention um, uh, the commercial of the genosensor, like the low cost, high sensitivity, and low limit detections. And um and for the conclusion of the article, discuss the significant threat that viruses um, has to the are to the humans, and how due to their unique non-cellular structures. Mm, um, even the PCR, it's accurate, has limitations such as the possibility of false positives and negatives, um, demonstrated during the COVID-19 pandemic, and the document highlight the need to develop biosensors that overcome the differences of the conventional detections methods.
Perfect. 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 Mm, so I guess every one of my team has participated. This is the paper that we got to review. Okay, so if anyone has any doubt, please feel free to just open the report that the doctor shared on the chat. As you could notice, that we skip another type of viruses. Okay, so please just get a read on this. It's really important. And we have concluded. Thank you very much. Thank you to my team as well. Bye bye.
Profe, ¿hay algún problema si presentamos el nuestro en español? O bueno, algunos. Es que yo me pongo muy nerviosa si hablo en inglés y me trabo. Y pues no, no puedo.
So, our article is a review of electrochemical henosensing of circulating biomarkers. This review is an approach of the different biomakers and henosensing development of techniques for the detection of different biomakers, viruses, and some other things. I don't know. So the importance of henosensing is the management of prognosis of disease. Give me a second.
So
Currently,
yeah, you
yeah,
can. Anything you want to continue with this?
yeah. Sorry, I was closing my door.
Yeah. Okay. Now you can. Yeah.
Yeah. So currently, electrochemical henosensors are considered a very promising analytical tool for the purpose of the fast response at low cost of the detection of some diseases. So the actual development of nanomaterials have a high impact on these henosensing techniques. Now, with these nanomaterials, the henosensing is a growing research field that is impacting the early diagnosis and treatment of any diseases. So we are going to continue with circulating biomakers. How we are going to explain something about that.
Am, ah, bueno, ay, profe, no sé si puedo decirlo en español. ¿Sí? Mira. Bueno. Am, a partir de esta parte, bueno, el inciso 2 nos habla más a profundidad acerca de los biomarcadores circulantes, que pues nos dice que son biomoléculas que se pueden medir objetivamente en los fluidos, en los diferentes fluidos del cuerpo humano, tanto sangre como orina, como semen, como leche materna, el líquido amniótico, entre otras cosas, igual por ejemplo el sudor o la saliva. Y pues dice que cualquiera se puede tomar como una medida, por así decirlo. O sea, que no importa si es normal o anormal, ya que pues son representaciones pues, de las funciones celulares de las células. O sea, te dicen si está bien o si viene como con un tumor o cosas así. Am, eh, También nos menciona un poquito en dónde podemos encontrarlos, que es bueno, que es en moléculas pequeñas, proteínas o exosomas o células tumorales, exactamente por los 9 que pueden ser anormales y todo esto. Y creo que es un poquito más abajo. Bueno, nos habla un poco de la clasificación. O sea, nos dice que existen 4 tipos de diferentes biomarcadores. Están los de detección de riesgo, que son los que se utilizan para la susceptibilidad a enfermedades de un individuo. Luego están los biomarcadores de pronóstico, que son los que nos da como el porcentaje de enfermedades el porcentaje de la enfermedad en un individuo el cual no ha sido tratado. Luego está el predictivo, que pues nos dice pues tanto la sensibilidad como la resistencia hacia cierta terapia. Y después está el diagnóstico de seguimiento de enfermedades, que pues como dice el nombre, nos ayuda al diagnóstico a seguir pues el, el progreso de la enfermedad. Y pues creo que es como lo que más importa de esta parte del artículo.
So for the third one, the electrochemical ay, no, no, gracias. Genotyping for the circulation biomarkers. Hold on. So on this part, we're talking about like potential applications of the electrochemical nucleic acid biosensing. Well, this technology utilizes sequence-specific profiles, biorecognition elements, and well, it's also been being related to biomarkers associated with various diseases, including pathogenic, genetic, emerging infectious, and non-communicable diseases like cancer and cardiovascular diseases. It also talks about the integration of magnetic beads, which helps in electrodes like screen printed electrodes and nanomaterials. Those magnetic beads improve sensitivity, reproductivity, and assay turnarounds by providing effective solid supports. And well, the bright side on this is that it's low cost and it's mass produced. It contributes to probability, miniaturization, and low sample consumption, making them suitable for like point of care tests. It also uses nanomaterials, which we are familiar with, like graphene oxide, mesoporous nanomaterials, nanoparticles, gold nanorods. And well, the review of this is it's kind of recent. On the article, it mentions that it was for the past 5 years, and it's the method of determining circulating biomarkers in liquid biopsy samples.
In the section 2.2, they showed two different genosensors, in which the objective of them is to detect some neurodegenerative diseases. And the first one was an electrochemical aptasensor, in which they want to sense prions proteins that are responsible to encephalopathies spongiform. And these proteins or prions are a group of, well, they do a group of neurodegenerative diseases, and one of them is the famous Creutzfeldt-Jakob, in which the brain of the person that is infected with this gets its brain spongiform. So to make this genosensor, they immobilize a specific haptamer in streptavidin, and also then they prepared an immobilization of polyidiomine with a G4 dendrimer supported with multi-wall carbon nanotubes that were absorbed in the surface of our electrode. They also then attached a redox marker that was ferrocene, and well, they made a cyclic voltammetry of the ferrocene, in which they measure the peak current, and then they determine the linear detection of the biosensor, and it was 1 picomole to 10 micromole, and the sample that they used was blood plasma. Next, this one. The next one, a biosensor, was an electrochemical detection system in which they use mesoporous materials with graphene. They use electroactive molecules like blue methylene and ferrocene. They use DNA probes. They attach it to the micropores of the material, and they made some electroanalytical signaling measure. When to the well, this was the objective to do that was to determine the acceptance of a target, DNA target, and those DNA probes change its conformation when the target DNA was attached, and with that, the porous will close, and they actually change the ratio of the electromolecule actives, such as the methylene blue, and they get liberated, and the current that they measure change. And also the last biosensor was quite similar to the second one, but in this one they use a polyprotein that was associated with Alzheimer's disease.
The next one is electrochemical genociding of viral infection biomarkers, and it's to employ a peptidic sequence of, no, to employ a specific peptidin sequence or specific DNA to detect electrochemic, genetic specific from some virus or antibodies. This part talks specific of three studies. One from Makisen et al. That made a sensor, electrochemic sensor, biosensor, I think, to detect the VIH. The next one is Oliveira, that they made a biosensor that can detect the serotype 3 from the dengue virus. And the last one talks quickly about a highly sensitivity impediment that can detect the norovirus in humans, that this, I think, provoke gastroenteritis, no, acute gastroenteritis, I think. And that's the most important of all the part.
Thanks. So in 3.4, you're seeing the electrochemical health sensing of bacterial infection biomarkers. This part is important because we can detect most common bacterial infections like Escherichia coli, Streptococcus pneumoniae, Streptococcus mitis. In this case, we can modify the electrode to improve the selectivity and the specificity of the electrode, so we don't have errors in this case. We have more common bacterial diseases like Pseudomonas inhivalis, that is causing periodontal disease, and schistosomiasis, something like Mycobacterium, and tuberculosis. Tuberculosis now is a bacteria that is highly difficult to detect and is a difficult part to work due to the infection and the risk that it implies. So developments like this can be a really helpful tool to detect this type of bacterial diseases. So the successful the results in this case is a very promising detection system. So can be applied to some others like leishmaniasis or leishmania bacterial disease that are very risk bacteria for a human body. For all this, we can see a better detection and specificity of the electrodes to detect the bacteria. So I think it's a good tool to improve and get a better result in the hospital, or something more easy to get for the patients. So general considerations for the
finally, the article provides information on the latest advances in electrochemical genetic detection by circulating biomarkers also known as biomolecules. And some of the circulating biomarkers that have been developed are to detect cancer, neurodegenerative diseases, bacterial and viral infections. Mm, uh, Conventional electrodes such as gold and gold electrodes prepared by photoli- photolithography, or these possible electrodes are used. Uh, Use of different bioreceptors such as linear and hairpin DNA, and there have been advances in this area that incorporate nanotechnology, with nanostructured surfaces has been developed with graphene and graphene oxide, and different bioreceptors are used for signaling, and the developed genosensors detected proteins and genetic biomarkers in the ranks of 0.5 picometers to 1 nanometer.
Um, in challenge to address and prospect for the future, says that health systems are looking for tools that are inexpensive, fast, non-invasive, and easy to use to obtain multiple results from a single sample, even in remote location, in order to take improve decision. Also say that genosensing electrochemistry has gained importance in clinical analysis by allowing the determination of circulating biomarkers in a non-invasive manner. Um, um, Although they are numerous biosensor electrochemical based in nucleic acid with the necessary sensitivity and selectivity, most of them remain in prototype development stakes, and only a few have demonstrated practical applications in real samples, such as serum, urine, and saliva. The transition of electrochemical genosensor from the laboratory to the market phase challenges, such as ensuring reproducible quantitative results, and that they are easy to use for non-expert users. Mm, Although the challenges, electrochemical genosensor are expected, are fast, sensitive, and easy to use, to have significant impact on disease diagnostic and monitoring in the near future. Finish.
Okay, thanks all. We're done. Yeah, so someone have a question or something like that? Comments? No, nobody. Hello. Hello. I am the Thanks, team. Thanks for the support.
Perfect. I think everybody has understood what is genosensor now, right? And that to electrochemical geosensors. I hope everybody has understood this well. Is there any doubt in this? I think it was well explained, because genosensors, if I would have taken one whole class, one session, I have the presentation ready. And I just had a discussion with Dr. Juan de Dios also, and he also suggested me that this can be a good exercise that the students can do and discuss and read. And so in that process, you can have a proper discussion. So anybody is having any doubt in this?
No, for the moment.
And did you understand? Thank you, Aranza. But anybody else, everybody could understand this well? Okay, fine. Now in 5 minutes, we will just gather some information. All right, I'll just So guys, I think you're all aware of everything. And now this is just an overview what we have to do. Nothing else. It will be very rapid. So I think everybody knows what are electrochemical sensors, and especially genosensors. Genosensors, when you talk, then it is with respect to DNA, right? And the genes responsible to interact with the electrodes. And here you can see the electrodes are there, and there are enzymes interacting with the electrodes, and the substrate interacting with the enzyme to get a product. And then you apply a voltage, and then based on the interaction with the enzyme, there is electron transfer reactions happening. Electrodes will receive the electrons, and then and then there will be a change in the potential, and that change in potential can be measured in terms of current. So this is happening this is the basic principle of electrochemical biosensors. And you know that there are three electrode systems. One is reference, other is working, counter electrode. So these are the three electrode systems which are responsible to do the measurements. And working electrode will have the proteins attached to it, or the enzymes attached to it. Basically, that is the place wherein you are doing the detection of the desired molecule. That's it. So these are then how you can create an electrode. On the surface of the electrode, you can create some chemistry. All right, you can have this kind of. So this is the electrode, and then there can be a dodecyl dimethyl ammonium bromide things. So you can either place the enzyme directly, or you can have this self-assembled monolayer kind of. So there is a self-assembled monolayer, and then have the enzyme, so that the enzymes are cleaned, and the transfer of electrons happen perfectly onto the electrode. So this particular reaction, this particular thing is only for the maintenance of this electrons to be moved in a proper channel and get into the electrode. So that's the motto behind this. And then the electron transfer reactions can be studied using this oxidation reduction peak of ferri and ferrocyanide. So this is the way where people can understand what is happening in the electron transfer reactions and how the
Profe,
sorry.
y las palabras están encimándose. O bueno, no sé si soy yo o si a todo el mundo lo ve, pero la parte de arriba está como encima. 12.
Usted lo puede ver de arriba.
Ajá. No sé si.
El problema es un problema aquí porque
Ah, ok.
tiene pequeñito problema aquí. Ya, porque tiene dos cosas aquí. Okay. So you can see here. This is cyclic voltammogram. Okay. No, esta. ¿Qué es esta? Perdón. Solo de cosa es que yo explico. All right. So, de cosa es que aquí es una cyclic voltammogram, ¿ok? Donde tiene, usted puede utilizar un tipo de enzimas, ¿ok? Enzima desde, es catalase peroxidase enzima, ¿ok? Y enzima es desde Mycobacterium tuberculosis. Y está integrada, de esta enzima es integrada adentro de fosfatidilcolin. Fosfatidilcolin de este tipo. So, por ejemplo, usted puede utilizar, ¿qué es esta? Es self-assembled monolayer. ¿Ok? Es monocapa de autoensamblada. So, the enzima catalase peroxidasa está adentro de fosfatidilserine, este tipo de fosfatidilserine, ¿ok? Está integrada. Y después, de todo este tipo de complejo es integrada en electrodo. Y de todo electrodo está en solución, que donde tiene ferrocyanide y ferricyanide. Eso es de plan, eso es de cosa aquí. De todo electrodo, de electrodo que tiene enzima y también que tiene de enzima con fosfatidilcolin, este cosa. Todo está integrada de todo electrodo y después está adentro. So the whole electrode is inside a solution which contains ferricyanide and ferrocyanide. That's the plan. That's the idea. And then when you perform cyclic voltammetric analysis, you will find that this is the behavior of reduction and oxidation of iron. Fe plus 2 and plus 3 will be formed, and the peaks can be seen, and it confirms that there are both the kinds of peaks which are existing, and the electron transfer is continuous, is happening in this particular reaction. So this is like the enzymes, and you can see here that the enzyme glucose now in this case glucose oxidase is the enzyme, and this enzyme will be interacting with whom? With glucose, which is a substrate, and then it will form like H2O2. That will be the reaction. And the transfer of, so now here here what happens is, sorry, so you can see here that there is the electrode, and this is the enzyme, and this is self-assembled monolayer. But on the top of this self-assembled monolayer, we will be keeping one mediator also. So whatever reactions happen, it will be via mediator. Okay, so mediator will be so what will happen is, whenever there is a electron generated, this electron will be transferred properly between, so the transfer of electrons should take place from the enzyme. Once the enzyme is interacting with the substrate, electrons are generated. This electron should not move anywhere else. It should move directly onto the electrode. So what is happening is the electrode, the electrons will move via mediator. All right, and it will move via mediator. So mediator is very localized mediator. It will capture the electron and transfer the electron. The job is it will capture the electron and transfer the electron onto the electrode. So this is the job of mediator. So here we have a mediator. Okay, this is known as ferrocene mediator, and glucose will interact. So you can see here, so you can see a mediator shuttles electrons between the enzyme and the electrode. All right. So what is happening is, you can see here, this is that. So this is the cyclic voltammogram. All right, this is a cyclic voltammogram, a huge peak, okay, with a high current. And so what is happening here? So there is glucose. The electrode is made up of glucose, enzyme, and mediator. When all the three comes together, then the electrons will be localized. Okay, so a mediator will motivate the electrons to move towards the electron. All right, towards the electrode, and then it moves towards the electrode, and then the electrode is responsible to have so there you'll find the reactions to happen perfectly, and electrode will capture the electron in a perfect fashion via mediator here. All right, but if you just have without a mediator, okay, without a mediator, if you perform the reaction, there is no transfer of the there is no good transfer of the electrons. Or if you just take a mediator and not the glucose or whatever, then again, there is no much good transfer of the electrons. So this is the reason electrons, the mediator with the enzyme and the substrate, all of them together onto the surface of the electrode, that will help so whenever the electrons are generated, that will be directly transferred to the electrodes, and then you can see this kind of reaction happening. Oh, sorry, this is not the one. All right, and when you talk about genosensors, now I have to explain you what are genosensors. So wait a minute. So when you talk about genosensors are also DNA, wherein DNA comes into picture. And DNA is responsible to interact with, they will be interacting with ruthenium or rhenium or any mediator. Okay. And these mediators are responsible to interact with the DNA. And now I want to understand whether, so it will be having only one single strand DNA. All right. And when there is a sample DNA coming, then that single strand DNA will interact with a sample DNA, all right, which is also a single strand. They will come together and they will form a double strand, and accordingly there will be change in the potentials. And that is what we will be like, that is what how the sensing, which you have already discussed, which you have explained me, how genosensors work and how the potential changes depending on the potentials, depending on the mediators which you have taken. There can be many mediators. You can have ruthenium, you can have rhenium, you can have even mediators which are like methylene blue. Methylene blue is also a very good mediator. And even so there are intercalators which will sit inside the DNA. All right, so if there is a double strand DNA, the intercalator will sit in between it, and then you will measure what is the potential. So this is how genosensors work, and how DNA detect. I mean to say, the glucose detection is done, and double strand formation is happening, or any biomarker detection, cancer biomarker detection is done, or any biomarker which is existing is done based on based on this either DNA or enzymes or antibodies. Even antibodies, whenever they interact with antigen, the antigen antibody complex is studied. Apart from that, the antigen antibody complex, you will be putting some ruthenium or any of the photo, like electroactive molecule, so that it can be transferred to, whenever then an antigen antibody complex is formed, then this complex needs to transfer the electrons to directly to the electrode, and the electrode will measure what is the status. Like the electrode will detect that change in the behavior, all right, in the change in the behavior. So what is the change in the behavior? When there is antigen alone, or the antibody alone attached to the antibody alone attached to the electrode. All right, this is one state. All right, you will have one cyclic voltammogram. But if this antibody is interacting with any complex, all right, any antigen, all right, and this antigen antibody complex is formed. Once that they are formed, along with any ruthenium or any electroactive material, they will be interacting with it, and then after that there will be change in the state of oxidation or reduction, and that change in oxidation or reduction can be detected with the change in the current. In this fashion, you will be able to detect whether there is a formation of whether there is any molecule which is being attached to the antibodies, or if there is any molecule attached, or any DNA, single strand DNA coming from the sample is attached to another DNA, or there is any enzyme interacting with the substrate. That can also be detected using this for this electrochemical studies. I hope you have understood this, and was it a good exercise for you guys, or it was like a worst exercise only for this thing? Because I was aware that you guys have been taught electrochemistry well in your system, in your so tomorrow also we will tomorrow I'm not going to have this kind of exercise, because tomorrow we are dealing with chemo sensors, and it's not just electrochemical biosensing. It's not electrochemical genosensing. It will be different. It will be chemosensing, wherein we will be talking about fluorescent molecules and etc. other things. So in this process, there is no electrochemistry in that. So that's the reason that I would like to teach you tomorrow, but not do this kind of exercise tomorrow. I'll be teaching you chemosensing tomorrow in my session. All right. So, but will this exercise could help you, or it was the worst exercise ever, guys? Now you have to speak a little bit. I need to understand. Don't put just this thing. Maybe Dennis Ramos can tell me something about this.
I'm sorry, can you repeat the question? I don't
My
listen
question is,
understand.
was this exercise good for you? The things which we had done today?
Yeah, I think it was a good form of participate more in the class, and we also have to learn about all the different forms that we can use this.
So it was a overall better. Or you can give me a clear feedback. There is no problem. I would like to listen to this, because this kind of exercise I have done for the first time. I was not aware of it, but let's see. I thought that okay, let's employ this, and whether you guys can enjoy this aspect and moreover, I wanted that you guys get into the concepts well, and there should be a proper interaction amongst you all, so that you will you might have some dialogues, because basically what happens is I only speak and you listen, and in that process I don't understand whether you have really taken up the subject well or not. But that this is the reason I had this exercise today, and I wanted to get an idea that whether you're really understanding the concepts well or not. And I think you have explained well, all of you. You have understood whatever you read. I think you have understood well, and there was no problem at all in understanding things. Okay. From my side, I was satisfied, but I wanted to see whether you are satisfied yourself. And if you feel that you're not satisfied, you can tell me that no, I didn't feel like it's a good exercise. We should not do this kind of exercise in future, in any time. Yeah, that's what I want to understand. So, okay, guys, I hope you can give me your feedback anytime, and and we can meet tomorrow again and talk about chemosensors. Okay.
Okay.
Okay,
Okay.
thank
Perfect.
you.
Thank you, guys. Thank you, and have a
Thank
nice
you.
time. And we'll discuss tomorrow. Bye. Thank you.
See ya.
Thank
Bye.
you.
See you.
See you.
Bye. See you. Bye. Take care. Bye.